Hello and welcome to part 2 of the chapter on cell physiology and pathophysiology. We left off, we had just finished the cell transport as the first topic, and then we were moving into cell signaling as the second topic in this chapter. You know this, cells signal to each other using a variety of molecules. Examples are neurotransmitters, hormones. One of the most important things that I want you to understand about this is that many of the current medications that we use exploit what we know about cell signaling. For example, the medications may mimic the signal, such as the case with levothyroxine, thyroid hormone, insulin itself, injectable insulin. Insulin's a signal, and so the medication is mimicking that signal. Those signaling molecules are either hydrophobic or hydrophilic or hydrophobic. That's going to impact where the receptors for those signals have to be located. Remember, we said hydrophilic molecules cannot pass through the cell membrane easily. So a receptor for a hydrophilic molecule better be on the outside. Whereas if the hydrophobic molecule is a signal such as a steroid hormone, that can be on the inside of the cell. Again, things that you probably already know. The autonomic nervous system manages a lot of our maintaining homeostasis by affecting different organs, glands, blood vessels. Mostly the neurotransmitters that are used are norepinephrine and acetylcholine or acetylcholine. Where with the endocrine system, it's also producing signals, we call them hormones in this case. And these are signals that are put into the bloodstream and then they affect cells that are far away. How a cell is going to respond to a signal depends on what receptors are present in that particular cell that we're talking about. So the signal may be inhibitory, that is going to be stop a process, or it may be excitatory, make something happen more. And it depends on what's happening at the specific receptor. I'll give you a very specific example. The messenger epinephrine. It is released by sympathetic nervous system stimulation. And there are lots of receptors around the body that are going to respond to epinephrine. A receptor referred to as a beta-1 receptor, these are found on heart muscle cells. They're going to respond in a way that's excitatory. When beta-1 receptors bind with epinephrine, it causes the heart muscle cell to contract harder and stronger, or faster and stronger. In contrast, same signal, epinephrine, but this time when it comes in contact with what's referred to as a beta-2 receptor, these are found on the smooth muscle cells in the bronchioles of the lungs. And actually, that's how I remember which one's which, is because I have one heart and I have two lungs. Yes, I shouldn't tell you these things, but this is how I remember things. The beta-2 receptors, those, when they're stimulated by exactly the same signal, epinephrine, their response that they elicit is to relax, tell the smooth muscle cell to relax and chill. It's not time to do anything right now. That causes bronchodilation. So the airways get more open and air can pass in and out through the lungs more easily. So that's what I mean by whether you get an inhibitory response or an excitatory response. It depends on what receptor are we talking about in what cell. Where the receptors are located, as I said, whether it's hydrophilic or hydrophobic, the signal that tells you where the receptor has to be. So for hydrophilic molecules, that is most of them, the receptors have to be on the cell surface. They have to be on the outside of that phospholipid bilayer. Because they're on the outside, but the effect you want is on the inside of the cell, there's this cascade of reactions that ends up happening to get to the response that we're after. And it turns out that that enzyme cascade, those different steps in the enzyme cascade, are also excellent targets for drug development. That's how many more modern drugs work, is by tapping into that signaling pathway. I'm going to throw out two terms for you, but I'm not going to go into a lot more detail. So the two most common types of these surface receptors are referred to as G-coupled receptors, and there's also tyrosine kinase receptors. Our textbook goes into excruciating detail on those two different things and the different pathways. We're not going to go into it in that much detail right now. But let me put things in context. We were just talking about those beta receptors. Those are both G-coupled receptors. It's a particular kind of receptor that passes the message into the cell through a variety of reactions. All right, and then going back to the signals, if the signal is hydrophobic, and the two things that fall into that category are thyroid hormone and steroid hormones, those things will pass right through the plasma membrane. And so the receptors for those things are located inside the cell. The thing that they most commonly do is bind to DNA and affect whether a particular gene gets transcribed or not. So it takes a little bit of time to get that reaction. So receptors for hydrophilic signals, there are various kinds, as I mentioned. I'm going to show you some diagrams, but I promise that we're not going to go into lots and lots of details. But I want you to get this general idea that when the signal binds, when the hormone, whatever the signal is, when it binds, it starts a cascade of reactions inside the cell. For now, we're ignoring the details of the cascade. So here is a G coupled receptor. Here's the signal molecule coming close to it. Once the signal molecule binds here, that activates the G protein, and these subunits go on and do something else. And here's another diagram. Here's another G-coupled receptor. Its signal is a different molecule. It bound here, and it activated, and it got something to happen with cyclic AMP. A third G protein activated by its own signal, it activates a slightly different kind of cascade, a different set of reactions, and its effect is to release calcium from the endoplasmic reticulum. My point here is what they all have in common is that we have a signal that's outside of the cell that cannot pass through the membrane, but we want an effect that's inside the membrane. And the way we get that effect is by stimulating a receptor and triggering this cascade of reactions. The other big main category of receptors are these tyrosine kinase receptors. They're beautiful and complicated, and we're not going to go into the details. Why I wanted to mention them is that things that control the cell cycle, remember we talked about G1 and S and G2 and go into mitosis, and okay, so that's all regulated. And sometimes the signal that tells a cell that it's time to make copies of itself, it's time to move on through this series of steps, that's related to these tyrosine kinase receptors. And sometimes things go wrong in the cascade in a way that even without the signal arriving, the cell starts marching through the cell cycle and starts making copies of itself. We call that cancer. And so understanding the signaling pathway and what happens in those reactions can help us figure out what kind of cancer, what's the mistake exactly, and how can we short circuit that, get in the way of that. And so these cascades, they're known as signal transduction pathways, and these are the targets of some newer medications, especially cancer drugs. So moving on, receptors of the hydrophobic signals, as we mentioned, those hormones, steroid hormones and thyroid hormones, they can pass right through the cell membrane, and they're in all the way into the nucleus in some cases, where the receptor is just waiting for them to arrive. Here comes one of those things. Ligand just means something that binds to something else. You'll hear it pronounced ligand or ligand. It's the same thing. When it binds to its receptor, that thing moves over to something else that's already on the surface of the DNA at a particular gene. And that can trigger transcription of that gene to happen. So you make the messenger RNA that gets sent out so you can make the protein. You do that transcription and then translation. And wrapping up signaling, we talked about extracellular signals, that some of them are hydrophilic, some are hydrophobic. This is the money part of this for you guys. Many of the medications that we administer to patients and that you'll be prescribing to patients affect the signaling process in some way. I just threw out here 4 main ways that I can think of, by acting as the signal itself, like thyroid hormone or insulin, by interfering with the production of a signal. If the signal we're talking about is angiotensin, then if you interfere with the production of angiotensin, like with an ACE inhibitor, an angiotensin converting enzyme inhibitor, then you're messing up, you're interfering with the signaling. You can block the reception of a signal with beta blockers, for example, or if you want to block the effect of testosterone for somebody who is a trans woman and wants to block the effect of testosterone, then you need something that's going to block the testosterone receptors. You can enhance the receptor of a signal. Some psych meds, benzodiazepines, for example, work in this way. Okay, so as you know, there's lots of drugs that you know, and what I want to point out is that lots of them have a mechanism that's related to cell signaling. All right, and that sums up the cell signaling part. Now we're going to move on to the third topic in this chapter, which is muscle cells. Again, it's a big change of gear. They're contractile cells. They're weird in this regard. They get shorter. That's the one trick they have, is they can shorten. The 3 main types, the skeletal muscle cells, cardiac, and smooth muscle. The first 2, everybody knows those. Smooth muscle is the one that gets people confused. Think about any hollow organ that you have, or a passageway, like the bronchi, bronchioles, hollow organ, like a uterus, a bladder, even the urethra has smooth muscle in it. Especially important are the arteries have smooth muscle in them. The way that muscles contract is by forming cross links between proteins that are inside the cell called actin and myosin. The actin's more passive, the myosin grabs a hold of it and pulls and gets the whole thing to be shorter. Just do that this way, it's shorter. One of the things that allows the myosin to finally hold on to the actin and start this process is having the level of calcium inside the cells start to increase. And that's enough to trigger the myosin to hold on to the actin and pull. Every time it pulls, you have to pay for it. It's ATP dependent. That's why exercising burns calories, because you're using ATP every time. Okay, it turns out thinking burns calories too, because we're using lots of ATP. All right, so we say the muscle cells specialize to shorten. We move the body around, as well as changing the volume and pressure of things to move air and fluid through the body's cardiac muscle and smooth muscle. Both striated includes skeletal and cardiac muscle. It means it looks like stripes under a microscope, but smooth muscle has some of the same structures. It's just not organized the same way, so it doesn't show up exactly as light and dark stripes. But they both do the same thing. They have cross-linking between the actin and myosin, and then break and reform. And then as it ratchets, it gets more and more overlap between those proteins, and the muscle gets shorter and shorter and shorter. Requires calcium to be present, and to pay for all of this, we need ATP. All right, diagrams. I'm not going to go into huge amounts of detail. What I want to point out is the myosin and the actin. The myosin's represented here in red, and the actin is represented in blue, and basically those two things overlap with each other and they slide past each other as the myosin kind of crawls along. Some terms that you might see, the prefix sarco refers to muscle. The cell membrane of a muscle cell is sometimes called a sarcolemma, and the smooth endoplasmic reticulum where the calcium is stored, like we don't have enough terminology. Instead of just calling it the smooth endoplasmic reticulum, they call it the sarcoplasmic reticulum. It's the same thing. It's the smooth ER inside a muscle cell. It stores calcium. All right, here's close-up myosin heads. These are the bits that kind of hold on to the actin filaments, and they can pull. There's some detail about how calcium has to bind to something. Basically, think of it this way. Actin is covered up. Where the myosin has to bind to the actin is covered up until calcium is present. Once calcium is present, then actin is available for myosin to ratchet onto. And then once we're done with the contraction, we put that calcium away, put it back into the ER, and then the muscle will relax. Oh, and here's that diagram showing that. All right, so there's the head of the myosin. You have to pay it. The presence of the calcium allows the myosin to bind to the actin, and then to relax, we have to put that calcium away and pull that myosin head back off. And that allows the muscle to relax. And there's the sarcoplasmic reticulum. This is inside the muscle cell. This is the cell membrane on the outside. And here's some myofibrils. These are the organelles that have all the actin and myosin in them, and allows the cell to contract. In smooth muscle cells, it's not organized quite so much. So it's still actin and myosin, but it's not lined up like this. So when we talk about calcium channel blockers, we're talking about medications that block those passageways that let calcium go through, but the passageways that let calcium go through on the cell membrane. So that's going to impact the smooth muscle cells, especially the smooth muscle cells in arteries and the cardiac muscle cells. It does not impact the skeletal muscle cells. And that brings us to the fourth topic in this chapter, that is cell adaptation, cell damage, and cell death. Cells can respond to stress by adapting. And in the next slide, I'll go over the different trophies and the different plasias. Like trophies, I mean hypertrophy and atrophy, and plasias, metaplasia and dysplasia. Then cell death. This is actually important part of growing and developing and maintaining homeostasis. So it's not always bad when cells die. The process called apoptosis or apoptosis, if you're British, that is kind of planned cell death. The cell has a set of steps that it goes through when it die in an organized way. And that helps to remove unneeded or unwanted cells, but it does not activate the inflammatory response, which is key. Now, examples of when this happens is, for example, early in development, there are a bunch of cells that get made that you can think of as kind of scaffolding. So when the limb is first being made, it's kind of a mitten. And so there's cells that join the fingers. And so and they're there on purpose, kind of to grow the blood vessels and grow the muscles that you need. And then finally, when kind of that part's done, then those cells kind of die off, and then the fingers can come away from each other. And so we lose the webbing as planned. Red blood cells don't last forever. And so aging red blood cells kind of have this process that they go through when they've reached kind of the end of their useful life. And intestinal epithelial cells, we expect that to happen and the cells just slough off. It does not create inflammation. In contrast, sometimes cells get injured severely and they don't have an opportunity to undergo this very organized process of dying off. This results in cell death that does cause inflammation. We call this kind of cell death necrosis. And finally, nutrient insufficiency, that is starving, can lead to cell death by what's known as autophagy or autophagy. So auto is self and phage is eating, so self-eating. The cell components get broken down and recycled for use by remaining cells. This is actually what is being initiated when people fast. They go an X period of time without bringing in more food. This is the big benefit of fasting. And we don't need to go into like whether fasting is good or not, but this is what people are trying to trigger by restricting nutrients. Okay, this information you can find on page 105 of our text. Here are the different pathies. So normal cells here in the middle, if we work them a lot, we need them to grow a lot, they get bigger. This is known as hypertrophy, or we can make more of them. This is called hyperplasia. If we do both things, then we get more big cells. Let's see, if we have disuse, something's not being used, the easiest example is muscles that are not being used a lot. So muscles that are being exercised, we have muscle hypertrophy, we get big and strong. And when we use them less, either because we're busy and we don't work out, or because there's paralysis or somebody's in a cast, the muscles atrophy, they get smaller. There are cases where cells are being exposed to a chronic irritant, and we end up having the cells, we make more cells and we make more cells that aren't quite right. We call this metaplasia. An example of where that happens, or several examples of where that happens, for example, in the airways of people who smoke, the pseudostratified columnar epithelium that has the cilia on it, that stuff dies off and gets replaced by some cells that there's multiple layers of them, but they're not very good at getting the gunk out of the lungs. You'll see this also in the esophagus of somebody who suffers from GERD, from gastroesophageal reflux. When the esophagus is chronically exposed to that low pH stomach acid, the cells change and they start looking more like intestinal cells than like esophageal cells. This can be an early sign. It can lead to esophageal cancers in the downstream if it's not addressed. One more place. People who are exposed to HPV infections have sometimes cervical cells that don't look quite right. We call that metaplasia, or when things go on long enough, we may end up with dysplasia and the actual development of cancer in the cervix, for example. All right, so those are different challenges and adaptations that cells have. Okay, so response to acute ischemic injury. The most common kind of cell injury is hypoxia, going without oxygen. Why that is a problem is because we are absolutely dependent on oxygen to perform oxidative phosphorylation, to do aerobic respiration. Remember the mitochondria, and the mitochondria is the one that makes the ATP, or where we make the ATP. We absolutely require oxygen to take electrons at the end of the electron transport chain. So when cells go for any length of time without oxygen, the ATP production decreases. So there's an obstruction, for example, somebody's having a heart attack. There is decreased oxygen available. The electron transport chain grinds to a halt because there's nowhere to go with these electrons. And so we're just dependent on doing glycolysis. You can get a little bit of ATP from that, but we're talking about 2 molecules versus 38 molecules of ATP. It's a big difference. So what happens? Decrease in ATP, let's point out what happens from that. Sodium potassium pump. can't pay for that, can't pump the sodium out. That's going to cause sodium to build up inside the cell, which is going to draw water into the cell, which is going to cause acute cellular swelling. What else happens? ATP is low, can't do the electron transport chain. So now we're dependent on just glycolysis. We do lots of that. That gives you a buildup of lactic acid. So things start getting more and more acidic inside the cell. Oh, sorry, let's go back. We had the sodium potassium pump. Oh, things gotten swollen. Those ribosomes, they pop off of the rough ER and you're not able to do protein synthesis. The other thing, you're doing glycolysis a lot more, you're building up lactic acid and the pH is going lower and lower, you're getting more and more acidic. Finally, the calcium pump, remember you had to pay for that calcium pump with the ATP. We're not able to do that. Lots of things happen, but most notably, we end up with damage to phospholipids, damage to the cell membranes and to the organelle membranes. And remember, one of the organelles we said were lysosomes, that were like the little stomachs. Yikes, when those get damaged and you release that stuff, that's going to start dissolving things inside the cell, and the cell just auto digests and gets destroyed. This is not programmed. This is not by design. These are just the consequences of not having enough oxygen and not having enough ATP as a result. The cells die by this necrotic process. And so that results in cellular necrosis. So to summarize, cells die a variety of ways. In necrosis, the cells swell, rupture, spill, initiates inflammation. If it's organized and programmed, it's apoptotic cell death. The cells shrink, little pieces break off, non-inflammatory, the immune system clears that stuff out. And finally, with autophagic, some cells can digest their own contents and provide nutrients for other cells. That's also non-inflammatory. And that is the end of part 2.